Welcome back to the first night of Soapbox Science Sydney's live online events. I'm Laura McCaughey and I'm your host for this evening. I would like to acknowledge the Gadigal people of the Eora Nation upon whose ancestral lands UTS's City campus, from which we are streaming, now stands. I would also like to pay respect to the elders, both past and present, acknowledging them as the traditional custodians of knowledge for this land. Soapbox Science Sydney is taking a different format this year thanks to COVID-19. Last year, our event was held in person at Circular Quay, where we had over 600 people question, probe, and interact with our researchers while they stood shivering on their soapboxes. Although we've had a change of format, our events still have the same goal at heart, which is promoting dialogue between the public and researchers about topics that are important. On this note, if you have any questions during tonight's webinar, please type them into the Q&A box in your Zoom control panel, and we will do our best to answer all of them over the next 40 minutes. You can ask questions anonymously, and also if you like a question someone else has asked and would like it answered, then please use the upvoting tool, which is the wee thumbs up symbol next to the question itself. This session will also be recorded, but will not be recording any audio or video input from the audience. If you have any concerns or questions over this, you can contact the Soapbox team via email at soapboxsciencesydney@gmail.com. Our brilliant speaker tonight, Nisha Duggan, is now a fully fledged Sydney-sider, having moved here to start her chemistry PhD 3 years ago at the University of Sydney. The focus of Nisha's research is to develop new therapies for stroke, and she's doing this by making a component of funnel web spider venom that has been shown in mice to reduce the extent of brain damage that occurs after stroke. Extracting venom from funnel web spiders to make medicine doesn't sound like the safest route, so I will now hand over to Nisha so she can tell us all about her role in mitigating this dangerous step.
Thanks, Laura, for that introduction, and happy National Science Week. I'm super excited to be here tonight, and so thanks so much for tuning in. So as Laura said, I'm doing my PhD at the University of Sydney, and I work for Professor Richard Payne in the School of Chemistry. And specifically, I'm a synthetic chemist. So I'm making new molecules in the lab. So that means I sort of fit into this white lab coat, safety glasses, pouring things into big flasks, like trying not to start a fire, mad scientist kind of a vibe. And as Laura said, the main goal of my research is to develop new drugs for stroke. And sometimes these come from some pretty unlikely sources that I'll get to a bit later. So first of all, I want to give you a little bit of background about stroke, what the current drugs that we have available are, and then later on, I'll tell you about the cool new drugs that I'm trying to make. So I'm sure that I don't need to tell you how terrible stroke is, but I did want to share with you a few statistics that I find quite shocking from the Stroke Foundation. So around 60,000 Australians will have a stroke every year, leading to around 10,000 deaths. This means that stroke kills more women than breast cancer and more men than prostate cancer. And beyond this, around 90% of stroke survivors will have some kind of permanent disability from the brain damage that's caused by stroke. And this could be a really minor or really major physical or mental disability after a stroke. So even if you survive a stroke, there still could be some really serious long-term consequences. So what's happening in the body when you have a stroke? So stroke happens when there is a loss or interruption of blood flow to the brain, meaning brain cells don't get oxygen. This lack of oxygen sets off a cascade of reactions in the brain that leads to brain cell death, which can cause disability. There are two main types of strokes. One is caused by blood clots or fatty deposits that block blood vessels and stop blood from getting to the brain. Around 90% of strokes happen in this way. Lifestyle factors can increase the risk of having this kind of stroke, but it's important to note that there are also genetic factors at play. So stroke can affect people of all ages and lifestyles. The other main type of stroke is caused by blood vessels rupturing. And this can be caused by high blood pressure or even from blunt force trauma to the body, which is causing blood vessels to rupture. Both kinds of stroke are extremely serious and can lead to death or permanent disability. So what are the drugs that we currently have available? So people who have been identified to be at a higher risk of having a clot-induced stroke for whatever reason may be put on blood thinners or anti-clotting agents like warfarin to try and prevent clots from happening. The problem with this is that they can have some really bad side effects, like excess bleeding and bruising. This makes sense because we know that our body needs to be able to form blood clots sometimes, like when we hurt ourselves. So if you take drugs to stop blood clotting, you are going to stop the so-called bad clotting that's associated with stroke, but you're also going to stop the good clotting that your body needs to heal. So what we need is a drug that can balance blocking the bad clotting, but also allowing the good clotting to happen. The other kind of drug that we currently have available is called tissue plasminogen activator, or TPA. This drug works to break down a clot that's already there. So this drug is often given to people who have had a clot-induced stroke right after they have their stroke to try and break up and dissolve the clot that's there. The problem with this is that this can only be given to people who've had a clot-induced stroke. If you were to give this drug to a person who had had a ruptured blood vessel kind of stroke, it would be extremely dangerous and could lead to excess bleeding. So clearly, the drugs that we currently have available are not sufficient. The blood thinners that are on the market have bad side effects, and there are no drugs to treat the second type of stroke caused by ruptured blood vessels. Also, all the drugs that we currently have available target clotting in some way. Maybe what we need is a drug that works in a completely different way. And I'll get to that a little bit later, and now I'll answer any questions.
Thanks, Nisha, for a really fascinating introduction into stroke. I didn't actually know that there were two different types of stroke. I've heard of the blood clot one, but I hadn't heard of the rupturing blood vessels. I have a question, a personal one. I vaguely remember learning at some point in my undergraduate that for the drugs that you give when you have a blood clot, you have to take them as quickly as possible after having the blood clot. Is that true? And if it is, why?
Yeah, so that's referring to TPA, tissue plasminogen activator. So it needs to be taken as soon as possible after you have a stroke to break up the clot and try to get blood flow back to the brain. So there's actually a really short window of time that you can have this drug. I think it's about 4 hours. And this is obviously a huge problem because first of all, like I said, you need to make sure that the person who's had a stroke has had a clot-induced stroke, not a ruptured blood vessel. So the stroke patient will need to be taken to hospital and have a CAT scan or something like that to confirm that there is a blood clot. And this poses all kinds of other problems as well, because people in regional areas who have had a stroke won't be able to get to hospital necessarily in time. So it's not really the best treatment option that we could have.
Yeah, that does sound like we're very limited as to what you can treat people with. Is there a way to prevent stroke? Lifestyle factors, exercise? Is there anything you can do to try and help yourself?
Yeah, so there are lots of things that you can do to try and prevent having a stroke. Number one is a healthy diet and lifestyle. So low fat, low cholesterol diets, and not drinking too much, not smoking, and being physically active. So that's sort of your best bet, really. Not that I'm a medical doctor, but...
And I'm sure that'll cure a lot of diseases as well. So
yeah,
maybe I'll get up and go a run in the morning. On that note then, so I've heard stroke only affects fat people and old people. So is that true?
So no, while the lifestyle factors that we've just mentioned can help to prevent a stroke, there are uncontrollable genetic factors at play. So things like specific heart conditions, or some people have high cholesterol conditions and high blood pressure conditions that you can't control. And actually stroke affects people of all ages. I think I saw a statistic that said about nearly 65% of stroke patients are of working age. So it's definitely not a disease that only affects old people or fat people.
Okay. Someone has asked, I know someone who's taken aspirin to try and prevent stroke. How does that work?
Yeah, so you might think of aspirin as sort of like a painkiller that you take when you have a headache, but actually it's also an anti-clotting agent. So aspirin blocks an enzyme called COX-1, and this enzyme is known to promote inflammation and pain, but also blood clotting. So if you take aspirin to block this pathway, it would be, so that's working towards preventing clots from happening. So it's one of those preventative treatments.
Okay. Um, and is there anyone else working on stroke drugs in Australia or around the world? It sounds like there's a great need for them.
Yeah, there are actually lots of people working on stroke, like making new stroke drugs, or also studying the other kinds of strokes. So I know at RPA they have a stroke study going on, and there's a Stroke and Heart Foundation in Sydney that's also working on, and there's people working on all different kinds of aspects of stroke. So not just the drugs, but also the rehabilitation after stroke and the genetic factors that would make you more prone to stroke. So it's definitely a big area of research, but we do actually need a lot of people to be researching it.
That's good. By the sounds of it, the more people working on it, the better. We've got a question here. Is there a link between stroke and heart disease? Because you hear the two of them mentioned together all the time.
Yeah, there are. So there are lots of sort of, I think, similarities in terms of risk factors. I'm not a medical doctor and I don't know too much about heart disease, so I probably won't comment too much because I don't want to say anything that's wrong, but there are definitely links.
Okay. In terms of time, we do have some more questions, but I'd like to hear a bit more about your research. So back over to you, Nisha.
Thank you for those questions. So now, before I start talking about the science too much, I want you to imagine that you are in a COVID-free alternate universe and you're at a barbecue with all your mates and it's summer and it's fabulous, but there's no bug spray. So you keep having to swat away all those mosquitoes and flies and annoying bugs. So I know this feeling all too well. Bugs absolutely love my blood for some reason. My housemates have even joked before that when we sit outside in the backyard, they don't need to wear bug spray if I'm there because the bugs will just go for me. And it's completely true. If I don't wear bug spray, I will always wake up with so many bites all over me. So one of the reasons that I always ended up with so many bites is that flies and mosquitoes and these kind of creatures have a range of chemicals in their saliva to help them gain what we call their blood meal. And this is an evolutionary trait. These creatures have evolved with the best body type and saliva composition so that they can suck loads of your blood really quickly to stay alive. So what kind of chemicals would they be? What would be useful for these flies to have and mosquitoes to have in their saliva to help them feed on our blood? Well, what these creatures want to do is to bite you and suck your blood for as quickly as possible without you noticing. So they've evolved to have chemicals like vasodilators, which are chemicals that can open up or dilate your blood vessels, blood thinners, and anti-clotting agents. So these kinds of chemicals are injected into your body when these bugs bite you, which is thinning your blood in the local area, preventing it from clotting, and allowing them to suck up as much of your blood as quickly as possible. So this is great for the bugs and super annoying for us, but maybe we could use these chemicals to our advantage. So remember that I told you that one type of stroke is caused by blood clots, and people who are at a high risk of having this kind of stroke may be put on blood thinners to try and prevent these clots from happening. But the drugs that we have currently available have bleeding and bruising bad side effects, which make them less than ideal. So what we have is a need for newer, safer blood thinners and bugs and mosquitoes that produce blood thinners in their saliva. But what's next? How do we go from a chemical in the saliva of a fly or mosquito that thins your blood to a drug that might be able to be taken for stroke? So first of all, biologists need to extract and separate and analyze the saliva. There's lots of different molecules in this saliva, so each one needs to be separated from the others and tested. Then, if one molecule is deemed to be interesting or exciting, a synthetic chemist might need to come on board to make it in the lab. As you can imagine, each tiny little insect can only make a tiny little amount of any molecule of interest. So you need a synthetic chemist like myself to develop a synthetic strategy to make this chemical in the lab. So in one of my projects, that's what I'm doing. We've identified a potential blood thinner in the saliva of a horsefly. And now I'm trying to make it on a bigger scale in the lab for further testing. This molecule is a peptide, which is a smaller protein made up of amino acid building blocks. So I need to find or make the building blocks and then put them together in the right order so that we can make a large quantity of this peptide for further testing. So we can't necessarily guarantee that this blood thinner will be safer than the ones that we have currently on the market. And there's still a really long way to go before it could become a drug. But I think it's a really exciting area. And I have at least partially been able to swap some of my annoyance for gratitude for those little creatures for producing these chemicals that might one day be life-saving. So I'll answer a few more questions now.
Thanks, Nisha. And I will definitely be inviting you to my next barbecue because I am the person who gets all the mosquito bites and everyone else around me is protected. So maybe if you're more tasty to the bugs, I'll get a break. So we've got a few questions here, and the first one is from Yi Lien, and she wants to know, and I think so would a lot of people, how do you extract saliva from a bug?
So this is the biologist's realm, but I think that they have like sheets of cloth and things like that they can get the bugs to like walk across, and they try and just like make them sting the cloth, and then when they sting it, the chemicals in their saliva will like come through, and you can collect that and then analyze it.
There you go, you learn something new every day, how to get saliva from a bug. I might have missed this, you might have mentioned it, but can your drug be used to treat both types of stroke that you mentioned earlier, or just the one type?
So this drug that I was just talking about will only be for clot-induced stroke. And it's really going to be like the idea would be to be a preventative type of treatment that you could take, if it's safe enough, to take long-term, if you've been identified to be at a high risk of having a clot-induced stroke, to sort of prevent the possibility of having blood clots that would relate to stroke. So this is still only the one type of stroke.
Okay. Are there other animals that have venom with these properties? What about snakes and stuff?
I'm not sure about, oh no, actually certainly snakes would, because they would be also sort of trying to bite things and like get things out of people. But definitely other bugs like mosquitoes are a big one, like ticks as well. Any kind of creature that relies on a blood meal from a host would probably be producing these kinds of chemicals.
Another question that we got in registration was how long until a drug like this could become available on the market?
Yeah, so this is sometimes a bit of a disheartening thing to think about, because the process from like discovering a potential therapeutic to it actually being on the shelf is actually super long. It could be like 10 years, because you first need to identify a particular molecule of interest and see that it has the effect that you want in the lab, in a sort of like, if you sort of like test it in little human body like systems, but in a petri dish, before you can actually test it in animals, then in humans, and so on, and then register the drug. So it's a really, really long process, unfortunately. So don't get necessarily too excited about seeing any of these drugs that I'm talking about coming out too soon.
Yep, I work on developing new antibiotics, and yeah, unfortunately, in the same boat.
Yeah.
We've got a question about side effects, or two questions about side effects. One is, what are the possible side effects of using a drug made from bugs? Could the person end up poisoned? And what are the side effects in general of using these blood thinners?
Yeah, so I'll answer the general side effects one first. So I sort of touched on this a little bit in the intro, but taking blood thinners can have some bad side effects because it could promote bleeding when you like cut yourself or things like that. If your blood's too thin and it's not clotting when it needs to, you could have these bleeding side effects, which are bad. So it is about finding the balance between the good clotting and the bad clotting. We're not sure about the drug that I've been just talking about. We don't know for sure yet whether it will have the right balance. We'll need to do those tests later. In terms of the side effects of taking a drug that's made from a bug, well, that also will need to be tested. But just remember that when you're talking about something that comes from a bug, you're not going to be having the entire suite of venom molecules that are in that bug. We would be separating them out and testing individual components to make sure each individual component that we might want to use is safe. So you're not going to be getting poisoned from taking a drug that's from a bug.
Yeah, and a lot of drugs that we take on the market today actually do come from bugs. About 40% of antibiotics that we take are actually produced by bacteria, for instance, and they have very minimal side effects as well. Somebody is asking if this medication that you're making will be given orally or intravenously, because TPA, which you mentioned before, can only be used intravenously and needs 24 hours of monitoring in ICU.
Yeah, that's a really good question. So it's likely that it might not be able to be taken orally because this drug is a peptide, and so peptides are smaller proteins, and we consume proteins in our diet, right? Which means that our body knows how to break them down. So if you were to consume a peptide drug orally, your body might break it down. So it might need to be taken intravenously or intranasally. Unless in the drug development side, we can work out a way to make the peptide more stable to all those things in your body that are chopping up proteins. So it's likely that it might need to be taken intravenously or intranasally, but there's the possibility with some sort of molecular engineering that it could be taken orally, which would obviously be the goal if someone was to be taking this drug regularly.
Yeah, I would definitely prefer oral over IV. I'm not very good with needles. Somebody else has asked that there are many, many different chemicals in bug saliva. How easy is it to isolate and identify which one it is that's active?
That's a good question. So I guess it depends on the way that the study is being done, because there's a few ways that you could be analyzing the saliva or something from a bug. You could be just taking the crude mixture and then testing it for different properties and then finding something that has an interesting property. Then it would be difficult to separate them all out and work out which one it was. The other thing that you could be doing is taking it, then separating it, then testing for interesting properties. So then you'll know which one is giving you the interesting property. And the other thing that biologists often do is to look at the genetic sequences for the particular peptides or whatever that you're looking at. And sometimes that can give you a clue as to what kind of molecule this would be and what it might be doing. So I guess it depends on how you're doing the analysis, but it can be tricky. But there are sort of lots of different little tricks that biologists and chemists can use to help them work out which is the molecule that they're interested in.
Well, thanks for that, Nisha. In terms of time, I want to hear more about your research, and then we'll have some more questions at the end. So back over to you.
Some really interesting questions. So just then we were talking about flies and mosquitoes. Now I want to talk about something that I think is far worse, which is obviously spiders. There's something that I just find terrifying. I think they're ugly. I think they're creepy. One of the worst memories I have from my childhood, or maybe when I was a bit younger, was when I saw this really weird looking spider in our house. So I bug sprayed it. And then I realized that the reason it looked so weird was because it was pregnant. So when I sprayed it, it exploded with spider babies that ran everywhere and completely freaked me out. And so I've hated spiders ever since. And I'm from Melbourne. So when I moved to Sydney, I was especially scared that I would come across a funnel web spider. I'm sure most of you know what they look like, but if you don't, Google it later because they are seriously scary looking. And not only are they scary looking, but they are incredibly dangerous. Funnel web spider venom acts on the nervous system. So it interferes with the way that your nerves send messages around your body. And this can be lethal. There have even been some reported cases of children dying from funnel web spider bites in just 15 minutes, which is just crazy. So funnel web spider venom is made up of a complex mixture of molecules. We know that some of them are really toxic to humans, but some of them might not be. They might have other properties that we don't know about. So our lab collaborates with a lab in Queensland that studies the venom of funnel web spiders and other creatures. So they extract the venom, separate it, and then test it for different properties. And recently they found a peptide from funnel web spider venom that has a super interesting property. So just to go back briefly, remember that I was telling you that when you have a stroke, you lose oxygen to the brain, and this sets off a cascade of reactions that leads to brain cell death. Let's think of this like a biological switch. When you have a stroke and you lose blood flow to the brain, the lack of oxygen flicks on the biological switch that causes brain cells to die. This is quite a complicated pathway that we're still learning about, but we do know that sometimes this switch can stay on for hours after you have a stroke, which means that even after blood flow returns to the brain cells might keep dying, which can lead to widespread, irreversible brain damage that can cause disability. And just to remind you again, we currently have no drugs to treat this. So jumping back to the peptide from funnel web spider venom, what our collaborators found was that when they gave this molecule to mice that had had an induced stroke, their levels of brain damage were reduced. And that's because this molecule can flick off that biological switch that causes brain cells to die after stroke. So this is a really amazing, interesting result, and could become a really important drug. But further testing needs to be done. So we need large amounts of this molecule. And even though funnel web spiders are obviously a lot bigger than flies and mosquitoes, they can still only produce a relatively small amount of any molecule of interest. So once again, as a synthetic chemist, I'm on board to try and develop a synthetic strategy to make large amounts of this in the lab, which I think, as Laura has mentioned, it would be a lot safer than trying to extract it from a funnel web spider forever. So once again, this molecule is a peptide. So I'm finding or making the different amino acid building blocks and putting them together in the right order so that we can get enough for testing. And the really key thing to note about this drug is that because it's not targeting clotting in any way, it's targeting the brain damage pathway that happens after stroke. This could be used for both types of stroke. So the idea would be that you could give this molecule to someone who had had a stroke, no matter which kind it was, and then their levels of brain damage would hopefully be reduced, and hopefully you'd be able to prevent high levels of brain damage and disability. So once again, there is still quite a long way to go before this could become a drug, but it is, I think, really, really exciting. There's no other drug on the market like this one. So I'm super excited to be part of this project. And I hope that if you, like me, are scared of spiders, that you might be able to change your mind at least a little bit about these creatures. So I'll answer some more questions now.
Thanks, Nisha. I don't know what I'm going to have a nightmare about tonight, either spider babies or one of my kids dying in 15 minutes from a funnel web spider bite.
Sorry.
But you did give us hope that you are looking at a potential treatment for this, what's currently an untreatable type of stroke. So that's a really exciting piece of research to be doing. We've got a lot of questions to try and get through. The first one is, can you give us some more insight into how you are testing these drugs to treat stroke?
Yeah, so the way that these drugs are tested, so this is actually done by the biologists in Queensland. So they have, there's a few stages of testing. So the first stage of testing is done in the lab, so it's not done on any like animals, definitely not on humans. So you sort of simulate these biological pathways that you have in your body, but you simulate them in the lab. So these are some of the things that are involved in these pathways are ion channels, which are different types of proteins. So you can set these up in the lab and then see how they respond to the drugs of interest. Then if you get promising results in that kind of experiment, you could move on to, you need to check the sort of toxicity and things like that before you go into animals. But if there's you get through those stages where it looks promising in the lab and then looks like it's probably not toxic, you can move on to these mouse models. And so they have a mouse model of a stroke where they actually give them, it's a little bit sad, but they give the mouse a vasoconstricting drug, which means that it stops blood flow going through a blood vessel. And then that sort of simulates what a stroke in a human would be like in this mouse. So now you have a sort of mouse that's had a stroke, and then you can see how it responds to the new drug that you're trying to test.
That's really interesting, and you can see why it takes so long for a drug to get to market with all the different steps that you have to go through. A few people have been asking, is it just funnel web spiders, or are there other types of spiders that have peptides that are similar?
Yeah, so there's lots of other, so there are thousands of spiders around the world, which each produce, millions of different peptides. So there definitely are other spiders that produce something similar. So specifically there is I think it's the Brazilian tarantula produces a molecule quite similar to this one from funnel web spiders. But the cool thing, which has been tested sort of in early stages for stroke research, but the cool thing about the funnel web spider one is that it's much more potent than the one from the Brazilian spider. So that's good. And hopefully that means that we'll have a better chance. And one other thing that I thought I would just mention about these spiders and these crazy things that they're producing, it might seem kind of crazy that they're producing these peptides that have these effects on humans. And I sort of like to think of it as like an evolutionary accident, because these spiders, they haven't evolved to hunt humans, and they haven't evolved to produce molecules that would have some kind of effect on humans. So but there are so many spiders producing so many different molecules that some of them just happen to have these like crazy effects on us, which I think is just totally weird and interesting.
We actually have a question about that. Why do you think the peptides are present in the spiders? You've just said that it's not to affect humans. So what do you think their purpose is for the spider? So I think that this molecule must have some kind of negative effect on some kind of prey of the spider. So we know that spider venom generally acts on the nervous system. So it acts on the nervous system of its prey as well. But the nervous system of so what a spider would be hunting would be like insects and lizards and that kind of thing. So the nervous system of like a lizard or another insect is different to the nervous system of a human, but there would be some similarities. So this venom molecule probably has like a really terrible effect on like a lizard or some kind of bug, but because our nervous system is different from theirs, it has a slightly different effect on ours. This is my own question, Would you need to know what effect that would have on the environment? Because a lot of drugs leave your system unmetabolized, so they leave your system as they went in and end up in the environment. So would we have to figure out what this molecule is doing so that we don't kill all the lizards?
That's actually a really good point that I haven't thought of before, but maybe we would. We don't want to be killing lizards.
It would just be interesting to know why these spiders have these peptides. So another grant that you can write is to figure out what these peptides are. This is a really interesting question that came through. If this drug stops cell death, could you use it to treat other diseases like Alzheimer's?
Yeah, that's a really good question. So I think that because this drug specifically works it works to prevent the cell death that's caused by low oxygen, it could be used in other diseases, but only in other diseases where there is a lack of oxygen. So it probably couldn't be used in Alzheimer's because the cell death pathway involved in Alzheimer's is different to the cell death pathway that's involved in stroke. But there are other diseases like certain types of heart diseases that have low oxygen levels that would be sort of flicking on that biological switch that I was talking about. So this drug could potentially be used in other diseases for cell death, but only where they're in environments of low oxygen.
And what other diseases are there environments with low oxygen? Can you give us any examples?
Yeah, so it's called ischemic heart disease, which just means low oxygen heart disease. Anything that's ischemic, because that just means lack of oxygen. So yeah, heart diseases, I can't think of any other. So I think potentially traumatic brain injury or other traumatic diseases, traumatic injuries where you lose blood flow to a certain part of your body and then therefore lose oxygen would be other kind of diseases where you'd be able to use this kind of drug.
Okay, that would be really interesting to take a side and see how applicable this drug could be in the wider market. Is this drug the kind of thing that would be stable enough to keep in an ambulance for a while? Because you want to give them drugs as quick as possible.
Yes, so it would be stable, probably at room temperature, probably for quite a while. So it would be a really great thing. So I guess the long-term goal would be that you would have this drug in all ambulances. Maybe you could even have it, like people could eventually just have it at their house or something, or even at a pharmacy, I don't know. But the point would be that it would be really accessible so that you could use it as soon as possible after a stroke. But what's also really interesting about the research that was done in Queensland was that they found that this drug was effective on these mice that had had a stroke up to like 8 hours after the stroke. It could still have a positive effect in terms of reducing the amount of brain damage. So compared to TPA that we were talking about before, that you need to have as soon as possible after you have a stroke, this drug, while you would still need to have it as soon as you could, there's a wider window where you could have the drug, which also means it's just a really good therapeutic option.
Well, that's amazing that you're doubling the time that you can wait to take the drug from what the TPA is. So that does sound really promising. We've got a question from Jason. He wants to know, are the tests primarily biological, or is there a way computer modeling could help?
Oh, that's interesting. The tests that are being done now are all biological. I'm sure that there's a way that you can. So in terms of computer modeling that I know of, I know that you can sort of model like a particular target in your body that you're interested in, and then you can also like model, so like get a 3D version of what your drug of interest would look like, and you can like study to see if they will like fit together in 3D, because often the way that drugs work is that they will bind to whatever target that you have because their 3D shapes like sort of match or whatever. So you can maybe predict whether a drug will inhibit a target of interest through modeling, but you do need to do the biological tests to be sure.
So in terms of this follows on from the modeling question, Once you're able to make this peptide, are you planning on designing, potentially with computer models, the analogues of this peptide to get greater or more specific effects?
So people will be making, probably I might make some analogues, or some other people might make some analogues, but we probably at this stage aren't looking at computer modeling. We'd be maybe more looking at ensuring that it's like really stable. So but I suppose computer modeling would actually be really helpful, but it's quite hard to do computer modeling on bigger peptides. So when you're doing computer modeling to help work out potentially better analogues, it's often on sort of smaller molecule drugs. I think it's a lot harder to do on peptides and proteins, but maybe it's something that I should look into more.
We've got another question from Yilien. Can we take small doses of the drug, like a vitamin or a nutritional supplement, to strengthen the brain against stroke?
I don't think that would really work in this case, because of the way that the drug is sort of specifically works to stop the cell death pathway that happens after stroke. If you're not having a stroke and you're not undergoing any of these cell death pathways, then this molecule probably won't be effective to have small doses of.
Well, unfortunately, we're out of time, or we're almost out of time. I have one more question that I'd like to ask you, which I think is the most important one, and that is, why did you get into research and science in the first place?
So I sometimes think about this question a lot, obviously. So I didn't always want to get into science. I was actually lucky enough to have two fantastic scientists for parents, but this actually nearly put me off doing science, and I nearly did like all kinds of other things like law, humanities. I didn't really want to do science. I can actually remember when I was little, almost getting annoyed with my dad for sort of debunking the pseudoscientific claims made in skincare ads, or when he told me that it was impossible that Abby on NCIS would be able to be an expert in all these different types of forensic chemistry, or that they wouldn't be able to get the lab results that quickly. So I used to think that knowing too much about science would sort of ruin the magic of everyday life. But then I studied science in school, despite all this, and I had a really fantastic teacher, my year 12 chemistry teacher, who was a woman with a PhD, and she was just so inspiring and great. So then the more and more that I learnt about science, the more that I thought that knowing about the science behind everyday life didn't actually take away from the magic, but actually added to the magic. Because science is just totally crazy. Like, there's so much mad stuff going on. Like, you know, the way the human body works, gravity, the way that the moon controls the tides, like, it's all just so crazy. But knowing about it and understanding why it does that makes it seem even crazier and cooler. And maybe I'm biased, but I kind of think that chemistry is the coolest of them all, because you're like manipulating different chemicals and putting them together to make new things with different functions. And to me, that's basically like the closest thing to real life magic that you could possibly do.
Well, we won't start a war tonight. We've got chemists, we've got microbiologists, we've got physicists, but we won't get into that. Thank you for a brilliant talk today, Nisha. Not so much for the image of spider babies. But thank you also to everyone who attended and asked questions. If you want to watch this talk again, or you know someone who would have enjoyed it, then we'll be posting it on our website in the coming week. You'll also receive a post-event email in the next 24 hours with an evaluation survey. Please fill this out, as it does help us gauge the success of our event and also make improvements for next year. We couldn't run Soapbox Science without our amazing sponsors, so I'd like to thank the University of Technology Sydney, Excite on Science, Equus, and the University of Wollongong for their continued support. We have many more great talks lined up over the next 3 nights, starting with Natalie Mattison in 15 minutes, talking about stress and mental illness. You can join all of these talks from the link you're on now, or you can find more information on our website, soapboxsciencesydney.com. Thank you again for attending and for asking such fabulous questions, and we hope to see you at our next talk.